I hope you can hear me well.
Yes.
And
yes, ma'am, yes.
Yeah.
I will start sharing my screen. All right, so probably some of you that you can see already my first slide of my presentation, you might already wonder, what is this pretty yellow flower on the picture of my first slide, and what it has to do with the title of my presentation? Well, I did not make a mistake and confuse it with Arabidopsis. The flower that you can see here, it's a parasitic plant called Strigolacton, and from this parasitic plant, Strigolacton got its name, because Stragolectans were first discovered as a germination stimulant for parasitic plants such as Striga. Those parasitic plants, they cause big damage to agriculture, mostly because they attack important or infest important crop species, such as sorghum, millet, maize, or rice. And Striga species are present in many countries worldwide, but the most damage are caused in Africa, West Asia, and also Mediterranean countries. So if a field is infected with one or more Striga species, the yield losses can be even up to 90%. And it is estimated that only in Africa, Striga species cost losses of $1 billion every year. So parasitic plants from Orobanchaceae family, such as Striga species, they developed special system to sense the host which is nearby, because they produce thousands of or 10,000s of tiny seeds that are present in the soil, and they can stay in the soil and wait for the right moment to germinate for decades. But because they are so small, they have also limited resources. So after germination, they need to connect to the host within a few days in order to survive. So to do so, they evolved the mechanism to perceive compounds that are secreted from the host plants. And among those compounds, the most studied one or the most known ones are Stragolectons. So upon perception of Strigolactons by parasitic seeds, the seeds will start germinating. They will attach to the host plant, to the root of the host plant, and create a specialized organ that is called haustorium. Through this organ, parasitic plant will withdraw water and nutrients from the host, and in that way weaken the host. And only after a few weeks, parasite will appear above the ground and will start flowering and shedding seeds. And these parasites are so detrimental for agriculture because, as you noticed, all the damage to the host plant is done, or a lot of damage is done before even the parasitic plants is visible above the soil. But now, very important question is, why do plants then secrete a compound or compounds that attract their enemies? So it's a kind of suicidal compound. And this question was answered only in 2005. So around 50 years after Stragolectans were discovered as a germination stimulant for the parasites. So it was shown by Akiyama and his colleagues that Stragolectans, they also induce hyphal branching of arbuscular mycorrhizal fungi. Those arbuscular mycorrhizal fungi, they live with symbiosis with 80% of land plants. And in this symbiosis, fungus, so AN fungi, will provide the plant with water and nutrients, mostly phosphate, in exchange for phosphoassimilates. So in phosphate-limited condition, plants will secrete Stragolectons, and those Stragolectons will cause germination of spores of IM fungi, but also secretion of fungal signaling molecules such as MYC factors, and as mentioned before, hyphal branching. And all these three processes are very important or crucial for establishment of good symbiosis between the AM fungus and the plant. Well, parasitic plants, they likely evolved they hijacked this signal between the AM fungus and plant and turn it into the germination stimulating signals. So to ensure that the host plant is nearby and that they can survive after germination. So there is a positive role of Stragolectons in the soil as a rhizosphere signal, because AM fungi, besides of helping plant to obtain water and nutrients, they also help plant to cope with drought and salt stress. They also increase pathogen resistance and resistance against heavy metal toxicity. But Stragolectans are not only rhizosphere molecules, but they also have a function in the plant, so the endogenous function, which was discovered many years later. But it was shown that Stragolectans inhibit shoot branching. So this is the most known role of Strigolacton in plants. And Strigolacton deficient mutants, so biosynthesis mutant, has many rosette branches in comparison, many more in comparison to the wild type plants. Okay. So after discovery of Stragolectin as a plant hormone, many other functions of Stragolectans were shown. So that includes root development, but also leaf shape, leaf senescence, and drought stress. So on the top panel, you can see all the phenotypes of Columbia, so the wild type plants, while the bottom panel, there are phenotypes of MAX4, which is Strigolacton biosynthesis mutants. So for instance, it is characterized by higher number of lateral roots, also by different leaf shape, which is actually a different ratio of length, of blade length and width. They have also delayed senescence, and they are less resistant to drought stress. So up to now, there are more than 20 Stragolectons that were discovered, naturally occurring Stragolectons. And they were isolated from different plants, but they all have some similarities in their chemical structure. And this includes the ABC lactonin tricycling ring, and then they are connected by enol ether bridge with the D ring. The R stands for the different substitutions, which are depending on the Strigolacton molecules. The connection between the C and the D ring is very important for the signaling and also for, mostly for the signaling. So what do we know about the signaling of Stragolectans so far? We know that Strigolacton molecule is perceived by the receptor, which is called D14 or dwarf 14. This receptor has a dual function. So it's something atypical for plant hormone signaling. Apart from being a receptor, this is also an enzyme that can hydrolyze the connection between the C and the D ring. And upon hydrolysis, the D ring only is covalently bound to the receptor, to the catalytic site of the receptor. And this allows the receptor to change its conformation. That change in conformation can further help for the interaction with other complex components, among which there is MAX2. It's an F-box protein. It's a part of FCF3 E3 ligase. And the E3 ligase, it's known to ubiquitinate proteins and target them for degradation. And apart from the interaction with MAX2, D14 also interacts with the repressors of Strigolacton signalings, which are called SMAX6, 7, and 8. So in the absence of Strigolactons, SMAX6, 7, and 8 repressors will interact with topless co-repressor proteins to deactivate or repress still unknown transcription factor or activity of transcription factor via histone modification. When strigolactons are present, then the complex is formed between the receptor, D14, E3 ligase with MAX2, and the repressor proteins. The repressor proteins, MAX6, 7, and 8, are then ubiquitinated and targeted for degradation by 26S proteasome. And in that way, they release repression of the transcription factor, which further leads to expression of strigolacton responsive genes. For now, only several of those are known, and those are BRC1, that is important for shoot branching, and TCP1, which is important for leaf elongation. However, as I just mentioned, that D14 is not the canonical or unique receptor for strigolactons among other plant hormones. Also, the repressor proteins, MAX6, 7, and 8, they are unique for plant hormone signaling because they have dual functions as well. Apart from being the repressor, they were shown also to bind DNA. So they're also repressive transcription factors. They can bind their own promoter regions, and in that way, they can create kind of negative feedback. So they can control their own transcription, providing another mechanism to control strigolacton signaling. So because although there are 20 strigolactons which were naturally occurring, they were isolated, but this isolation is quite expensive and difficult. Therefore, for research purposes, we use strigolacton molecule that is chemically synthesized. And the most commonly used one is GR24. However, it was shown that GR24 is not one molecule, but it's actually a racemic mixture of 4 enantiomers. And out of those 4, the 2 indicated in black are so-called natural strigolactons because of the stereochemistry and the connection between the serine and the D-ring. And those will be perceived by D14 and will trigger strigolacton pathway. But there are also these 2 indicated in blue. And these 2 enantiomers, they have non-natural stereochemistry. So they cannot be produced by plants. They're only a result of chemical synthesis. And it was noticed that actually those 2 compounds are perceived by yet another pathway in Arabidopsis. So it must be that they can mimic something else, so some other compound. And what are those compounds? Well, it was shown that the same pathway can be triggered by carychins. And carychin, it's a term, aboriginal term, which means smoke. So after a fire in the forest, carychins are produced from burnt smoked material, burnt plant material. And after the rain, they can be washed to the soil and induce seed germination of many plant species that are also so-called fire-following species. And this will help to reestablish the landscape after fire. So the carychins are not produced by plants, but they are produced from burnt plant material, and they help plant seeds to germinate. So up to now, there are 4 carychins that were isolated from smoke, and they share some similarities in the chemical structures with Stragolectans, which is the butanolide rings. Apart from sharing similarities in the chemical structures, there are also some components of the signaling pathways that are very, very similar. So carychins are perceived by Ki2. Ki2 is the receptor, and this is also alpha-beta hydrolase, and it's a close homologue of D14, so the receptor of strigolacton signaling. The E3 ligase, MAX2, is also a central player in carychin signaling. And the repressor proteins, SMAX1 and SMAX2, they belong to the same family as SMAX6, 7, and 8, so the repressor proteins from Stragolectan signaling. Although much less is known about the carychin signaling pathway, it is predicted that upon perception of carychins, again, the complex is formed between the receptor Ki2, MAX2 E3 ligase, and the repressor proteins SMAXs. So the repressor proteins can be ubiquitinated and degraded, releasing downstream responses. However, what are the downstream targets, which are the transcription factor or keratin responsive genes? It still remains to be discovered. Then something interesting was noted, that the Arabidopsis Chi2 mutant, so the mutant of the receptor, actually it also shows some developmental defects. So there must be another endogenous compound that is perceived by the same pathway. And for now, this compound is not discovered, but it's predicted to exist, and therefore it was named Chi2 ligand or KL. So, as I just said, keratin and Stragolectons, they have some similarities in the chemical structure, some similarities in the signaling pathways, because the receptors Chi2 and D14 are homologs. They're both alpha-beta hydrolases. There is the same central role for MAX2 H ligase in both pathways. And also the repressor proteins belong to the same family. But apart of this, they have different but also some overlapping roles during plant development. So keratins or Chi2 pathway, they're mostly involved in the early stages of plant development. And those involved seed germination in, let's say, specific conditions that I will explain later, but also early seedling development, so photomorphogenesis, while Strigolacton or D14 pathway, they're important for later stages of plant development, including shoot architecture, so shoot branching, and leaf senescence. There are also several plant developmental features that are for which both pathways are important, and they have interplay. And those include root architecture, leaf shape, and stress responses. So as you can see, it's very difficult to talk only about Strigolacton pathway without mentioning the other one. And the goal of my project was to discover what are the new players or novel other players in Strigolacton signaling pathways. So what are the proteins that are acting downstream of SMAX6, 7, and 8, or what are the proteins that are controlling them? So there is also this saying, show me who your friends are, and I will tell you who you are. And that's why we wanted to know what is actually the function of SMAX7 and what are the proteins interacting with it. And how did we do that? So we used tandem affinity purification in Arabidopsis cell cultures. So for that, we transformed cell cultures with SMAX7, so that was our bait, which was tagged with the double affinity tag consisting of streptavidin binding proteins and protein G. But before starting with tandem affinity purification, we wanted to be sure that it's a good environment actually to study Strigolacton signaling. So we treated cell cultures, MOC, or with GR24. We collected material at several time points, and then we tested protein expression. And as you can see, already after 30 minutes, we see some degradation of SMAX7, which is to be expected upon addition of Strigolacton analog. So this convinced us that cell cultures, they are a good environment to study Strigolacton signaling and to look for novel components of the signaling pathway. So how the method itself works. So we first use, so in the cell, we first extract the proteins, then we use IgG beads in the first purification step to pull all the SMAX7 together with the interacting proteins. So after several washing steps, we use rhinoprotease that will cleave off protein G. And then we can perform second purification step in which we use streptavidin beads that will pull again SMAX7 with the streptavidin binding proteins. And after several washing steps and elution, we have kind of pure complex of SMAX7 together with its putative interactors. And those were identified using mass spectrometry. So we got a list of putative interactions, putative proteins. And what we noticed from this list, that 5 topless and topless-related proteins were identified. Those are those co-repressors that were already known to interact with SMAC7. So that was another confirmation for us that, that our approach actually worked. But apart from finding those topless proteins, we also found several novel interactors. And in this talk, I will focus only on the 3 interactors that are marked here, which is FYPP1, FYPP3, and SAL4. And those are the part of protein phosphatase. So protein phosphorylation is one of the most studied protein post-translational modification in which the phosphoryl group from ATP is attached to serine, threonine of tyrosine of the protein, or it's by kinase, or is then removed by phosphatase. And protein phosphorylation can change protein structure, can change its localization or function. In Arabidopsis, there are 26 catalytic subunits of protein phosphatases, and they can be divided into 6 groups. And among those, based on their structure and function, and among those, there's the serine threonine protein phosphatase 6, and the catalytic, or PP6, and the catalytic subunits of this phosphatase is FYPP1 and 3, so those that we found. But besides of having catalytic subunits, this PP6 holoenzyme also consists of regulatory subunits. And in Arabidopsis, 4 such regulatory subunits were identified, and those are SAL1, SAL2, 3, and 4. So in our tab data of SMAC7, we actually found 3 components of PP6 holoenzymes. Those were the 2 catalytic subunits, FYPP1 and 3, and 1 regulatory subunit, SAL4. PP6 is already known in Arabidopsis to have some targets of PP6 complex are known. For instance, it is dephosphorylating PIN proteins, so it's involved in auxin signaling and in controlling auxin transport polarity in the root or leaf epidermis expansion. It was also shown that PP6 can dephosphorylate AB5, which is transcription factor important in ABA signaling that regulates seed germination. And besides these 2 hormonal pathways, PP6 can also dephosphorylate phytochromes, influencing flowering time, and phytochrome interacting factors, and in that way regulating photomorphogenesis. So we had this 3 putative praise, FYPP1, FYPP3, and SAL4. And first we wanted, of course, to validate if there are indeed real interactors of SMAC7. But as I already mentioned, it is difficult to talk about Stragolectan signaling pathway without talking about the Karyakin pathway. So we not only checked if those PP6 phosphatase components interact with SMAC7, but we wanted to also know if they interact with SMAC1. So first of all, we check their localization, if they localize to the same subcellular compartment, because this is already first indication of possible interaction. So what we know for SMACs is that both SMACS1 and SMACS7 are expressed only in the nucleus. And something which is very characteristic for them, that they're expressed in the nucleus not homogeneously, but into like kind of dots or so-called nuclear speckles. Then we also checked into subcellular localization of our putative praise. So for the catalytic subunits, FYPP1 and FYPP3, we could see that they are expressed in the nucleus and in the cytoplasm. We also cloned the N-terminal part of, which we call FYPP-NT. This is the N-terminal part of FYPP, which is identical for both FYPP1 and 3. Also, we had some problems with cloning or expressing SAL4. Therefore, we cloned only one domain, which is SASP domain of SAL4. But this domain was already shown in for mammalian PP6 that it's enough or it's sufficient for the interaction with the catalytic subunits, and also probably for interaction with the target proteins. And the SAS domain was also localizing to the nucleus and cytoplasm. So based on this, we concluded that SMAXs, they have some overlapping expression pattern with the PP6 complex components. Therefore, there might be an interaction between them. So in the next step, we perform these two hybrid analyses. And the blue color indicates interaction. So indeed, we could detect interaction between SMAX7 binding domain and SMAX1 binding domain, and the N-terminal part of FYPP. And this was quite strongly visible. There was also some indication of possible interaction with FYPP3, but not FYPP1. But this could be also due to technical limitations. For SAL4, we could not detect the interaction, but there was some interaction visible with the SAS domain only. So this was our first validation method. Then we performed film analysis. So we co-expressed SMAX7 GFP, which was our acceptor, together with the N-terminal part FYPP NT fused to RFP. And if these two proteins are interacting, or if they are in close proximity to each other, there can be energy transfer between the acceptor fluorophore, so GFP, and between the donor fluorophore GFP and its acceptor, so FYPP NT-RFP. And if this happens, so if they're interacting and there's energy transfer, then the average fluorescence lifetime of the donor, so SMAX7, will decrease. And indeed, this was the case, that when these two proteins were co-expressed, the average fluorescent lifetime was significantly lower in comparison with SMAX7 GFP only, or a co-expression of SMAX7 with nuclear localization signal RFP. And similar results were also obtained for SMAX1. So if SMAX1 GFP was co-expressed with FYPP RFP, the average lifetime fluorescence was shorter, which indicates interaction as well. And there we also noticed something very interesting, because as I already mentioned, SMAXs, both SMAX1 and SMAX7, they're localizing in the nucleus to this specific dot-like structures, which are called speckles. The function of those speckles is still unknown, but it is possible that they indicate kind of transcription active sites. So SMAXs are localizing to the speckles, but the phosphatase itself, so the FYPP1 or 3, if it's expressed alone, is just homogeneously expressed in the nucleus. But if we co-express the phosphatase together with SMAX1, so you can see a SMAX1 GFP, it's in the speckles, but then phosphatase, FYPP NT-RFP, also goes to the speckles. So the SMAX1 or SMAX7 as well, which you can see here, is kind of dragging the phosphatase to this nuclear speckles, so changing its localization, which for us was another indication that these two proteins interact. And similar observation was also already noted and described for the interaction between SMAX7 and D14. So SMAX7 can also change localization of D14 and drag it to these nuclear speckles, and the same with topless proteins. So we also tested the SAS domain, if it can change its localization and go to nuclear speckles as well. As you can see here, SAS RFP itself, it's only in the nucleus, but the moment when it's co-expressed with SMAX1 or SMAX7 GFP, then it also goes to this nuclear speckle. So it's co-localizing in the nuclear speckles. So after this several validation methods, we could conclude that PP6 complex, both catalytic and regulatory subunit, seems to interact with SMAX7 and SMAX1. So they may be involved in Stragolecton, but also Karakin or KL signaling. But what is then physiological role of PP6 complex? So I showed you already this scheme at the beginning that Stragolecton and Carycins are important for many developmental processes. But here we will focus only on 4 of them. So seed germination, hypocotyl elongation, lateral density, and shoot architecture. So for all the assays, all the physiological assays, we tested double mutant of FYPP1 and FYPP3, which is shortly called here F1, F3. The single mutants, they showed no phenotype, probably because of the redundancy of these genes. We also tested Cell4 mutant, but Cell4 mutant also didn't show any phenotype. And now we are busy with generating multiple order mutants. So we tested the double mutant, but we also created a triple mutant between F1, F3, and MAX2 to see if this double mutant can somehow influence phenotype of MAX2 itself. And the first assay, which is testing shoot architecture, we grew plants for 50 days in the normal day-night conditions. And after those 50 days, we counted number of rosette branches. As you can see on the graph, there was no difference between number of shoot branches of Colo and the double mutant F1, F3, while MAX2 mutant, as expected, show almost or more than double the number of shoot branches. The triple mutant F1, F3, MAX2 showed the same phenotype as MAX2. So here we could conclude that probably the PP6 complex is not important for shoot branching, or it's not involved in the control of the number of rosette branches. In the next assay, we checked for lateral density. So we grew our seedlings for 9 days on the plates with half MS medium. And after 9 days, we counted the number of lateral roots, and then measured the root length and divided the number of lateral roots by the root length. And what's known for MAX2 that it has more lateral roots, or the lateral density is slightly higher than for Colo. This is an extreme example, let's say, but this phenotype is also quite variable. So in our case, in our assay, we could see that indeed MAX2 had slightly higher number of lateral roots, or lateral density was slightly higher, but this was not significant. But what's interesting, we observed that FYF3 double mutant had significantly lower lateral density. And the same was the case for FYPP1, 3, MAX2, the triple mutant. So we think that the FYPP or the phosphatase may be involved in the control of lateral density or lateral root development. In the next assay, which is more related to the Kai2 pathway, we tested hypocotyl length of the seedlings that were grown for 4 days in continuous red light. And in this condition, in mock, you see elongated hypocotyl of Colo, which is shortened by if GR24 is present in the medium. And in agreement, MAX2 has a significantly longer hypocotyl than Colo, and it's not influenced by addition of GR24 because it's the signaling mutant. So what we could observe is that the double mutant FYF3 had the same phenotype, or there was no difference between the double mutant and Colo and the wild type. MAX2 had significantly longer hypocotyl length, and this was not changed in the response to the treatment with Strigolacton with GR24. But the triple mutant had slightly but significantly decreased hypocotyl length. So again, it seems like PP6 can be involved in hypocotyl elongation. And our last assay was seed germination. But seed germination in specific conditions, because Arabidopsis seeds, they can just germinate and they don't need any chemicals like GR24 or Carycins for that. But so to study Kai2, they put seeds in a buffer in 96 well plates in continuous dark, and also at higher temperature at 25 degrees. And then we check color will germinate lower, and this induction can be induced by GR24, while MAX2 will almost not germinate. And indeed, this was of Colombia in mock conditions was around 40%, and if GR24 was added to the buffer, it was increased up to 80 more. MAX2 showed almost no germination, and the same phenotype had a triple mutant of F1, F3, MAX2. And we also noticed that the double mutant F1, F3 had significantly lower germination in mock conditions, but was still induced by addition of GR24. So it seems that F1, F3, or the phosphatase, can be also important for seed germination. So in two assays that I just showed you, we noticed something interesting, and that was in lateral density assay and in hypocotyl elongation, that in the triple mutant, we can see suppression of MAX2 phenotype. And this is something that was also observed for SMAXs. So SMAX, the name SMAX, is actually means suppressor of MAX2. So MAX2 phenotype, for instance, in lateral density or elongated hypocotyl, can be suppressed in the in case of hypocotyl in SMAX1, MAX2 double mutant, or in lateral density can be suppressed by both SMAX1, SMAX2, and SMAX6, 7, and 8. So we thought maybe the phosphatase has something to do with the turnover of or stability of SMAX proteins. So does PP6 regulate the turnover of SMAX proteins? And to test that, we express SMAX7 GFP in color background, but also in the double mutant F1, F3 background. And then we treated plants with GR24 at several and collected samples at several time points. So what we noted that already in mock, the expression of SMAX7 was very, very low. So this could indicate that the stability turnover of SMAX7 is dependent of PP6. This is also quite a preliminary experiment, and we still need to confirm it. However, we were very happy to see a MAX2 independent mechanism of SMAX1 turnover. So we also would like to test whether whether phosphatase is important for SMAX1 turnover. And if this turnover is MAX2 independent, how it would look based on our phenotypes in lateral density and hypocotyl. Well, so to summarize, to the slide that I showed you in the beginning about the Strigolecton signaling, we can add some new components. So we could show that PP6 complex, phosphatase complex, is interacting with SMAX7. Whether its function is to dephosphorylate SMAX7, it's still under study. So we did some phosphoenrichment experiment to test actually if PP6 is dephosphorylating SMAX7, but at the moment I'm waiting for mass spectrometry results. It seems that PP6 can control stability or turnover of SMAX proteins, but whether this mechanism is MAX2 independent, it still remains to be determined. And also for PP6, we knew already that it's involved in auxin, ABA, and light signaling, so we can add another one, so strigolactone and keratin signaling. However, what is the exact role of PP6 in strigolactone and keratin signaling? It's quite difficult to say because the phenotypes that we observed were not always consistent. I showed you only a part of it. And why is that? Well, you can see PP6 phosphatase is already involved in auxin, ABA, and light signaling. So all the phenotypes that we also check for strigolactone and keratin signaling, such as root phenotypes or seed germination or hypocotyl elongation, it could be that what we see, it's not because of impaired signaling of strigolactons or Ki2 keratins, but because other signaling pathways are disturbed. So we still have to find out or find a way to prove or to to see which are the real, what is the real function of PP6 in strigolacton and carotene signaling that is kind of disconnected from the other ones. Okay, and with this, I would like to acknowledge some people. I would like to thank the Rhizosphere Lab, especially my boss, Professor Sophie Gormachtig, also our technician, Annick, and 3 students, Anse, Adne, and Dan. So these are the PhD students working on Stragolectons that I'm supervising. Also, I would like to thank the Interactomic Lab and Phosphoproteomics for their help with TAP and phosphor enrichment experiments, and Evelyn and Professor Daniel van Dam for Fred Flame experiments. I would also like to thank you for your attention, and I'll be happy to answer any questions that you have. And if you have some more questions afterwards or would like to stay in touch, this is my email address. Thank you.
Okay, thank you so much, Sylwia. It was very nice presentation. And I mean, you are addressing a very important question in Strigolacton biology. Sylwia, you can hear me? Okay.
Yes, I can hear you. I will stop sharing.
Okay, so those who has any questions, please write down in the chat box, or you can raise your hand and you can start asking. So Sylwia, I will ask you a question. So it's quite clear that Strigolacton has some role in phosphorylation. But did you already try applying GR24 in somewhere and just checking the phosphorylation status of global phosphorylation status of some protein samples or something?
No, we did not try that. We tried to do more dedicated phosphorylation studies. So what we were thinking is more than that the phosphatase can be involved in dephosphorylation of SMAXes. So this we checked, we wanted to check directly. So we expressed transiently our SMAX7 together with the phosphatase, with FYPP1 or without it. And then we were checking if there's some difference in phosphorylation. But we did not do the general phospho study, but I think it will be very interesting to do, because there's one option is that SMAXes are dephosphorylated by the phosphatase, but it can be also that because SMAXes are also chaperone proteins, so it could be that they're actually SMAXes are helping somehow phosphatase to dephosphorylate something else. So we were also thinking, like you said, to maybe check what are the targets of this phosphatase and which are Stragolecton or GR24 dependent.
Yes, Dr. Sylwia. So we just lost Sibu sir. He has lost his connection. He will be coming back to the session. So I just wanted to ask, why did you choose Strigolacton as such? What was the motivation behind selecting Strigolacton?
To work with, okay. So because so I started working on it in 2013, and there was really not much known about the signaling itself, because they were discovered already in 1960s as a germination stimulant for parasitic plants, but the role as a plant hormone was shown only, I think, in 2010 or 2008. So when I started, we knew that there is MAX2, and that maybe there is D14, which is the receptor. So I thought it's a very new field, and there are still so many things to be discovered. And also I like the fact that it's involved, it's not only plant hormone, but it's also involved in, it's also rhizosphere signal involved in many processes. So I think that was the most important for me.
Yes. So we have a question from Deepthi Suresh. She has asked, like, what are the emerging roles of strigolactons in plant responses to stress and development?
What are the
emerging roles of strigolactons? Emerging role of strigolactons in plant response to stress and development.
To stress, yeah, I'm not sure completely. I will not, because it's not my field of research, and this is actually quite new, that it's shown that Stragolectans, they're involved in stress responses, but not only Stragolectans, but there are these two pathways. So both Chi2 and D14 pathways are important for stress responses, and those are growth, as I mentioned, but I think there is also something with response to pathogens, or there is at least one paper about that, but yeah, I don't know details about it. It's not something that we studied. So I'm sorry for that.
Yes. So if you have any more questions, please type your questions in the chat box. You can also directly ask. Just raise your hands. We'll call you out. So we have one more question from Varsha KP. Majority of the scientific researchers are carried out in Arabidopsis plant. So why this plant is used as a model organism?
Well, for us, for instance, because so first I started working with Arabidopsis to know what is the function of Stragolectans as a hormone. But then I kind of changed to parasitic plants because this is more applicable. And we wanted to know how Stragolectans are, how the signaling works in parasitic plants. But parasitic plants cannot be used as a model system. So we still have to use Arabidopsis because, for instance, parasitic plants cannot be transformed. And they're also very difficult to grow. So there is no other way to study, or there are some possibilities, but it's very difficult to study directly, for instance, in the parasites, and that's why we use Arabidopsis. If it was more general question, of course, Arabidopsis, it's easy to use as a model plant because genome is known. It takes only 2-3 months to get to another generation. There are many molecular tools available already in these plants. But then, of course, there's a question of if it will be the same for all the other plants, right? If we work only with a model plant. I hope that answers your question.
So I think Sibu sir has a question, sir.
Okay, Sylwia. So if you treat some cell culture or something with the GR24, can we measure some difference in phosphorylation status? Did we? You must have already tried that one, no?
I think I was answering that question and then you were lost.
Yeah, so I just lost my connection, sorry.
No, we didn't try it, but I think it will be interesting to try. But what we did, it was more dedicated study to see if SMAXs are dephosphorylated by the phosphatase. But it would be very nice also to check that. But it's it's a more broad problem. It's a project for PhD student. It's by itself.
I mean, you can do
if there are some differences.
Yeah, you can do a simple treatment of some cell culture. You have this Arabidopsis cell culture there, then isolate the protein, global protein, and just do the phosphorylation status. Enrichment. So that's probably.
But what we would be more interested, I think, is to see in relation to this phosphatase. So, for instance, use seedlings like Columbia wild-type seedlings and the seedlings of double mutants FYF3.
Of course.
And treat both of them with GR24 and then compare the phosphorylation, which proteins are phosphorylated or dephosphorylated in relation to. So also more direct approach, because if we start from cell cultures or just from color, then we will get a lot of phosphorylated proteins. And because, as I said, the PP6 is already known to be involved in many hormonal pathway and other signalings. So then I think it will be more difficult to decide which is actually really related to Stragolecton.
And why do you have a differential developmental response? You have no effect in shoot branching, but you have an effect in lateral root development. I mean, root density. So how can you speculate the differential effect actually in development?
I don't know. So we still don't know why is it, and we still don't know if the effect that we see in the root, for instance, it's for sure because of impaired Strigolacton signaling, and not because of the influence on pin proteins, because it's known that Strigolacton and auxin signaling are interconnected as well. But it can be also that something that we still have to check where the phosphatase is expressed. So that's what I'm going to do. We know that it's expressed in the root for sure because that was published, but maybe just it does not have a role in the shoot branching, right? If it's not expressed there.
I think that would be the most likely thing you can go for. And I'm a spatial distribution of this particular protein in root and above ground and below ground organs. Now that could be the one thing what you can probably look. And also probably you may have to generate one more mutant combination. Maybe you can include one auxin mutant also crossed with your strigolactone, this P1, P2 thing. You know, in case if you are strongly suspect that you have some you have some crosstalk between auxin, probably that mutation may also help you, probably, isn't it?
Yeah.
Yeah. Okay. Okay, Sylwia. Nice talk. Thank you.
Thank you.
Yeah.
We have one more question in the chat box from Prajita Jayan. What is the role of strigolacton signaling and crosstalk with other phytohormones?
So what we know so far that there's a crosstalk with auxin signaling, as I already mentioned, in the root, but also in the shoot, that there is one pathway. So I kept it quite general in the presentation, that in the signaling, it is predicted that there is some transcriptional control, but there is also some non-transcriptional Stragolectan signaling. And it was shown that maybe Stragolectans can directly influence PIN, so auxin transport via influencing the PIN localization or switch from the top to bottom membrane, or I don't remember exactly, sorry. But there are some options. There is some crosstalk with auxin proteins. It was with auxin signaling. It was also shown that there is some crosstalk with cytokine signaling in the root development. But for now, there was another question, I think, about gibberellins.
Yes.
No. The only two pathways that I know so far, it's the auxin and cytokine signaling are the two pathways that the crosstalk is known. Maybe it will be still discovered, because as I mentioned, the Stragolacton field is quite, as a plant hormone, it's quite new, and there are a lot of papers, manuscripts coming.
Okay. So the next question is by Bijida. Do strigolacton have any effect on the plant that makes them? What is the effect of, I think she's thinking of what is the effect of strigolacton on the plant itself.
Okay, so Stragolectans are plant hormones, right? So there is and they were discovered, so there's effect on shoot branching, and Stragolectans are inhibiting shoot branching. So what is thought that they kind of help plant to cope with the stressed conditions. Like, for instance, if there is not enough phosphate, then the plant will not invest energy in growing more shoots. So Stragolacton will inhibit shoot branching, while will be exuded, for instance, to soil to attract arbuscular mycorrhiza. Yes, Stragolectans are also important for lateral root, for root hair length. So they have many function in the plant as well. But I don't know if I should mention like all the known functions of Stragolectans, or.
Yes. So next question is by Anu Balakrishnan. So is strigolacton have any pharmacological effect?
Actually, yes. So there are some studies. It's not, of course, my field of studies, but there are some manuscripts showing that connection between Stragolectans and cancer. So that they were tested if they have some effects on treatment of cancer cells.
Next question is by Shruti Srikumar. Can we regulate strigolacton biosynthesis by gibberellin signaling?
This I don't know, but from what I haven't read about it. So I don't know, sorry.
Yes. Okay, so any more questions to ask? Please ask the questions directly. Sibu sir?
Yeah, yes, please. So
I think
there is no more questions, no? So we can probably wind up. Sylwia,
yes.
Thank you so much, Sylwia. It was very
Thank you for
nice,
inviting me.
very yeah. Okay, okay. Then who is it? I think it's Varsha KP or Devika.
Yes, sir.
Okay,
So,
please.
thank you, ma'am, for the excellent presentation. We have Ms. Varsha KP to propose a vote of thanks. So, Varsha.
Okay. A warm good evening to all. On behalf of MacPass College, I feel immense pleasure to take this opportunity to propose vote of thanks to eminent dignitary Dr. Sylwia Struk, VIB-UGENT Center for Plant System Biology, Belgium, for taking out time from your busy schedule to grace the event. Your talk was so thought-provoking, ma'am. Thank you so much for such a wonderful presentation. I will further extend hearty thanks to our beloved principal, Father Dr. Charyanji Kotel, for providing the support to make this webinar successful. And my gratitude to our backbone, Dr. Jenny Jacob ma'am, head of the Bioscience Department. I'm also thankful to Dr. Shilpa Lakshmi ma'am for introducing our eminent speaker. My sincere thanks to our research director, Dr. Jyotish Kumar sir, and Dean Ms. Beena Charya ma'am. I take this opportunity to specially express my deep regards to Dr. Sibu Simon sir for your coordination. My sincere gratitude also goes to the organizing committee for conducting this webinar event. Also, my deep sense of appreciation and thanks to all participants for attending this webinar with great enthusiasm and made it more interactive. Once again, I thank you all for being with us in this evening. Have a wonderful day. Thank you.
Okay, thank you.
Thank you,
Okay.
Sylwia, for your presence.
Thank you.
Okay,
So-
then Sylwia. We'll be in touch, Sylwia. Bye.
Bye bye.